and welcome to this video in which we are going to explore how we differentiate among different Staphylococcus species. So we have these two cultures here, culture A and culture B. I know I did that backwards, but you get it. And these cultures are both look like this under the microscope after gram staining. So what do we see here? They're the blue-purple color, so that means they're gram-positive. And they're cocci and clusters, so we are going to go with the assumption that this is a Staphylococcus species. And clinically, this is super relevant because we need to be able to differentiate Staph epidermidis, which is normal, non-pathogenic skin flora, from Staphylococcus aureus, which, well, as we know, lots of people are colonized with Staph aureus, can be pathogenic. So how do we figure out which is which? We have these cultures, A and B. One is Staph aureus and one is Staph epidermidis. We have already talked about a couple of ways that we might differentiate Staph aureus from Staph epidermidis. So for example, growth on mannitol salt agar. They're both going to grow because they're Staph species, but what would we expect from Staph aureus? We would expect that color change, the yellow color change as a result of the fermentation of mannitol, whereas with Staph epidermidis, no color change, even though there would be growth. And how about with blood agar? What have we talked about with blood agar? We know that this is differential for hemolysis. And so with Staph aureus, typically we see beta hemolysis, whereas Staph epidermidis is non-hemolytic or gamma hemolytic. Now, we haven't talked about coagulase in lab, but you have heard about it in the bacterial virulence factors portion of the class. So we will look at this today, but you already know that Staph aureus produces coagulase, that's one of its virulence factors, whereas Staph epidermidis does not. The last test that I want to talk you through is one that we haven't explored much, so I'm going to take a little time. This is called a latex bead agglutination test, and this relies on the concept of antibody-antigen specificity. Antibodies are proteins that our body produces, part of our immune system, in response to specific antigens. And in the vaccines portion of the class, you'll hear about antibodies and the role that they play in memory and specificity. But the idea here is that specific antibodies bind to specific antigens. So they're like a lock and a key. And therefore, we can know if a particular pathogen or organism is present if we have an antibody that's specific for that particular organism. So these latex bead agglutination tests, again, antigen-antibody specificity, but also agglutination. One of the properties of antibodies is that they cause agglutination or clumping. So with this test, we start with a known specific antibody for Staphylococcus aureus. So we have this Staph aureus antibody, and it will bind only to this antigen on Staph aureus. Staph epidermidis does not have this same antigen. So if the whole point of this is figuring out Staph aureus from Staph epidermidis, this should allow us to identify if the culture is Staph aureus or Staph epidermidis. So again, we start with this specific known antibody for Staph aureus, and it's attached to these latex beads in these little clumps. So you have a latex bead and you have the antibody. And we are going to add our two samples, sample A and sample B. Now, if the sample contains Staph aureus, the antibody will bind to that specific antigen, and you will get agglutination or clumping. And so what we see, it's not like a giant clump, it's more like little blue freckles or flecks on the slide. If you see that agglutination, that means that you know that sample contains Staph aureus, because the only time agglutination happens is when that antigen is present to bind to the antibody that we started with. However, if when we add the sample, Staph aureus is not present, you still have those antibodies, because that's what you started with, but you have a different culture. Say this is Staph epidermidis. It doesn't have the same shaped antigens, which means that there's no binding, no agglutination, and no clumps. Now, in general, that would simply tell us that the sample contains something other than Staph aureus, but because in this case, we know that our only two options are Staph epidermidis and Staph aureus, we can say then that this sample contains Staph epidermidis. So in the next portion of the video, we are going to actually look at the results for coagulase and walk through the process of doing latex bead agglutination so that we can identify cultures A and B. Hi there. In this portion of the video, Amanda and I are going to show you the coagulase test results and then walk through doing the latex bead test and recording those results as well. So for coagulase, each tube contains rabbit plasma and the bacteria that we tested, and we started this earlier because it takes several hours to incubate. And we expect to see coagulation or clumping or solidification in the tube that contains Staph aureus, and no solidification in the tube that contains Staph epidermidis. So our results, Amanda, do you want to show the two results?
Yes. So we have these two tubes. Turn them around so you can see. A and B. Can you see those labels?
Yeah.
And you can't really tell just from looking at it like this, but if I tilt the tubes, see how this one is runny and this one isn't. Whoa, the big glob. That is coagulation, as opposed to this one, which is not. So you can record those results for A and B.
Now we're going to move on to our latex bead agglutination tests. So Amanda, grab out 2 slides and please label them A and B. And we're starting with our Staph reagent. So you can see it says Staph. And this contains the latex beads and antibodies that are specific to Staph aureus. So that's our known specific antibody. So give that bottle a little swirl. Don't shake, don't foam, but a little swirl. Awesome. And please put one.
So good at swirling.
You are an amazing swirler. 1 drop, please, on each slide. Now we're going to use your loop, so go ahead and fire up the Bunsen burner. And go ahead and flame your loop, because aseptic technique and always. And what you're going to do is you're going to add a little bit of each culture, the appropriate culture, obviously, to each slide. You're going to smear a little bit of the bacteria on the glass next to the drop. So we'll start with A. Good aseptic technique. Pinky. Awesome. All of these skills that we are becoming familiar with. And once Once again, she'll put the tube down so that she doesn't feel like she has 1,000 things in her hands. So again, smear the bacteria on the glass next to the drop. And then drag the drop with your loop over the bacteria to mix them. So get it nice and mixed. And you can flame your loop. And while you're doing that, if you're able, you can just sort of gently rock the slide back and forth. Sorry, I'm asking you to do 18 things at once. Why don't I do this while you flame your loop?
Thank you.
I know, bossing you around. Okay.
It's all good.
So I'm not seeing much here, but it can be hard to know what the results are if you don't have something to compare it to. So let's hold that aside and do the same now for culture B. Add a little bit of culture B's sample.
I just flamed my loop, so we're good.
Again, add some of that culture to the slide and then mix together. Get a nice mix.
And then would you like to
I would love to.
tilt that for me? Thank you.
So now, oh, already, do you see? You see
Actually,
right there? Is it better?
Reflection of the legs.
Okay.
There we go.
Those blue flecks and clumps, that is really clear agglutination. So it didn't need, sometimes it takes a little bit of time of mixing back and forth, but super obvious agglutination for culture B, whereas culture A, totally fluid, no blue specks, no agglutination, agglutination.
Catching all of that. There we go. Now we can see that nicely. Awesome.
Good?
So we have B in which hand?
B in this hand and A in this hand.
Awesome.